There are three different methods for construction of phylogenetic trees, which are distance-based method, maximum parsimony-based methods, and the third one is maximum likelihood-based methods. Maximum likelihood and parsimony-based methods are characters-based methods. On the other hand, distance-based method is used for continuous distances. So if I go into details, so what happen is for distance-based methods, we have common matrix which is UPGMA. Then we have neighbor joining method, minimum, and then maximum, and then evolution methods. And they are always used for distance measures between sequences, and these sequences should be of continuous characters. On the other hand, maximum parsimony and maximum likelihood method, ये दोनों जो methods हैं ये discrete characters को यूज़ करते हैं। अब हम कैसे डिसाइड करें कि हमने अपने जो सीक्वेंसेस हैं उनमें सिमिलरिटीज़ को या उनके एवोल्यूशनरी इन्फॉर्मेशन को फाइंड आउट करने के लिए कौन सा मेथड यूज़ करना है। तो उनके लिए स्ट्रेट सी गाइडलाइंस हैं। फॉर एग्जांपल, अगर सीक्वेंसेस जो हैं हमारे काफ़ी ज़्यादा सिमिलर हैं, देन वी शुड गो फॉर मैक्सिमम पार्सिमनी मेथड। बट द प्रॉब्लम विद मैक्सिमम पार्सिमनी इज इट्स वेरी टाइम इंटेंसिव, इट टेक्स लॉट ऑफ टाइम। अगर हमारे सीक्वेंसेस जो हैं वो मीडियम सिमिलर हैं, देन वी शुड use distance based method. They are actually fast methods, but again they are not very accurate. And finally, if we have very dissimilar sequences, then what we should use maximum likelihood method, but they are the most time consuming methods. So I will start from maximum parsimony. What is maximum parsimony and what is its different steps? So again, as I started, that maximum parsimony, it finds a tree that explains the data with minimum number of steps. This is the basic definition, and it predicts the evolutionary tree that minimizes the number of steps required to generate the observed variation in the data. So basically we want to capture variation within the data. What is that variation and how it is captured? So again, this is appropriate for similar sequences which are very closer to each other, and it's time consuming, and there are different software packages available for calculating maximum parsimony. For example, one common package is Phylip, which is basically phylogenetics for information for inferencing, and POP, which both use these information of maximum parsimony. So what are different steps for calculating maximum parsimony? So initial step is to find out most informative sequences or most informative sites. So how this information is find out? There is a generic rule. For example, if I see this example, which is provided in this table. So we have different sequences along the x-axis and y-axis. So what we say that along the vertical axis, we have different taxon or taxa, which are basically categories of sequences. And on the row side, we have sequence positions and characters. So what happen is that I have said that 1, 2, 3, 4, 6 and 8, they are not informative. On the other hand, 5 and 7 are informative. How I say that these are informative? So if I look into the 5 one, we will see that there are two characters which are repeated. So which are G and A, which have two occurrences here in this one and this one, 5th. And in the 7th one again, we have two G's and two C's. So for a site to be informative, it is necessary that it should have minimum 2 frequency, and again it should be repeated in the same column. So here I have chosen these informative sites as 5 and 7. So if I take an example of column number 5, where we had two characters G and A which were repeated for 2 times, how we can build a tree out of these sites? So 3 examples are given downwards, which are basically for using this column, which is 5th column, and we have two characters GG and then we have A और A. अब हमारे पास क्या है, जो फर्स्ट हमने ये वाला दिखाया है, इसमें what happens is that we have tree 1 which has this G information. These are the two first G's and these are the two A's. To make them in a rooted tree or unrooted tree, basically what we need is that we make A here and G here. So this G represent these two G's which are here, and this A represent these two A's. So and then we have a combination here, which this sign, it shows that we have only one length tree. Okay, the second one, it is basically combination of again those two G's which are shown here and those two A's which are shown here, and then we have A in common between these two, and then this tree is of length 2. And finally, the third tree, again, it is showing 2 Gs here. But now what we have done is that we again have these A's, and again, so the relationship or this length is again these two, length of this tree is 2. So this is how we make phylogenetic trees for fifth. Similarly, we can use the same information for the seventh one, which is shown on this diagram. You can see this explanation in handout also. So again, following the same steps, we have length 1, length 2, and length 2 trees for seventh column. Okay, so this is how we can make trees. Okay, so what are comments then? So it's very good if we have number of sequences very low, but as the number of sequences will increase, this tree building step will become very exhaustive. So for example, if we have 10 sequences, then we would be having at about 2 million of trees, which can be completed by those sequences. So it will be very time complex, and it will be a problem in NP complete, which is very tough to deal with. So what are the different comments then? We have different softwares available, such as Phylip. So what happens is that it is very good when we have only 11 to 12 sequences. Then we have DNA computation performs, basically it performs genetic phylogenetic analysis using compatibility criteria. So compatibility criteria means that it will have a tree which can handle very large number of sequences. And then we have protein parse, which is used for protein sequences parsimony trees.